the session will be from Dr. Sam Taylor and Catherine Walsh, because today is Fascination of Plants Day. So Sam and Catherine have kindly agreed to share something about their fascination of plants, their research, and generally their inspiration as to why they're interested in the subject. So as I say, feel free to share questions as we go along. I think that's most of my housekeeping notes. So I think Sam and Catherine are sort of going to introduce themselves, but just to say that they're researchers within the department here at Lancaster Environment Centre. And we have quite a lot of plant and crop science researchers here, many of whom have produced some online activities for today for Fascination of Plants Day. So if you want to go to the website, I'll share that link in the chat shortly. If you want to go to the website afterwards and get involved in some interactive online activities, then you can continue the thoughts about plants and how wonderful they are into the rest of your afternoon. So at this point, I will stop sharing. We will hand over to, I think it's Sam that's going to start today with an introduction to himself and his research.
Okay, yeah, thanks, Ali. Yeah, my brief for this was really to talk about why I'm interested in plants and what I'm doing. I'm not really going to talk about my research much in terms of the details today. I think I'll skim over that, and if people do have questions, they can bombard us with those. But where I started with plants was really as an animal population biologist. So my PhD was actually about aphids. I had some plants involved. They were potato plants, and we were interested in control of aphid pests on potato plants. But there was quite a lot of thinking about plants involved in this, because the subject of this was how a pest population responds to changes in the plant itself. So this is probably as detailed as I'm going to get in terms of scientific content here, but there's a diagram on the left of this figure that shows from some potato plants that I grew at the time how the tuber mass increases with plant development. So you're going through time since the plant emerged from the ground, and there's a buildup of leaves, and those leaves are used for photosynthesis, and then that's translated into reproductive organs in terms of the tubers. Now, there was an observation previous to my doing my PhD that aphid populations tend to increase suddenly and get to very high numbers in the early part of the season, and those often disappear later. And that had kind of been tied into the idea that something changes about the plant as far as the aphid is concerned somewhere as these tubers are starting to develop here. And from our perspective, we were interested to know whether the stuff that's being generated by photosynthesis or the sugars that's being translocated to make those tubers is changing the quality of the sap in the plant and how that's affecting the aphid. So I did these kinds of electrophysiology experiments where you take a series of aphids. There's one marked there. You can't really see it on the picture, but there's an arrow pointing at where it is. And it has a wire going from that aphid into an amplifier. So I could record how the aphid was interacting with the plant. And the expectation was that if these plants got too sugary, basically, then the aphids would find that difficult. That would be stressful for them, because they have to osmoregulate more if there's too much sugar in their diet. So the figure on the right-hand side at the bottom here is an illustration of what happens to your relative growth rate as an aphid population if you have different amounts of sugar in your diet. So basically, as you get to the right-hand side of that graph, some of these aphids start to suffer a lot. So yeah, this all got me thinking about what plants are doing at different times of day, how they're moving things around inside them. There's a really dynamic system there, and part of what we were interested in with the aphids was whether there was more stress at times of day when there was photosynthesis compared to times of day when there wasn't so much photosynthesis. As I worked on aphids for a while, I learned enough about plants that I was able to do postdoctoral research on plants. So I went from the University of York, where I was working with aphids, to University of Sheffield. And one of the reasons I went to Sheffield was because there was this wonderful postdoc opportunity that was going to send me to South Africa and Hawaii as part of my research time there. So you can see on the bottom left of my slide here is a picture of our field experiment that we had in South Africa. This was a comparative experiment where we had C4 and C3 grasses from a number of different lineages that we had in wet and dry treatments. And we were trying to understand, whether this thing called C4 photosynthesis, which is sort of often referred to as a turbocharger for photosynthesis, it evolved in a number of different grass lineages. And the question was, how different were those grass lineages in terms of what that turbocharger was doing to their performance? So at this stage, instead of just thinking about things moving around plants, I started to think about how efficient plants could be and what it meant to have the ability to get more carbon in through your leaves through photosynthesis, and how you use that relative to your nitrogen resource. And in the context of comparing species from different habitats, what time of year do they want to grow? How is the temperature different in different places? And so on. I went from comparing lots of different species in Sheffield to, in the center there, you've got a picture of me comparing lots of different genotypes of switchgrass. So I'm the sweaty person in the back. This is a summer day just outside of Austin in Texas. We've got a representative of the UT undergraduate cohort there with his hook'em horns cap on. And we had these gigantic rainout shelters so that we could do experiments where we were looking at how genotypes of switchgrass that had been brought from different ecological zones in the US compared with one another when we put them under different regimes of soil moisture availability. These tunnels obviously created a rain break, but they were also quite warm because the daytime temperature around there is about 38 degrees Celsius in the summer anyway, and so we all got to rehydrate quite a lot. Luckily, the kids in front of me there that are taking samples, they at least are working with liquid nitrogen to freeze their leaf discs, so that that's keeping them a little bit regulated. But you certainly learn to use sunblock, something I learned that plants need quite a lot of later in my career. But yeah, so there I went from thinking about how efficient plants can be to how efficient different groups of plants, but are basically the same species, but grow in different places and have adapted to those different places can be. And then the thing on the right is just to demonstrate that at some of what I do, we use techniques where we have to kind of build our own kit to understand things. So this thing that's shown on the right-hand side is a setup for measuring leaf hydraulic conductivity that I built when I was in Australia. And I had to kind of fettle together a series of computer programs to get a balance to read out and build a system that meant that I could keep my leaf that I'd cut off a plant happy with a supply of water and monitor how that supply of water was being used. So I've ended up doing a lot of different things with a lot of different skill sets over the course of my career to understand how plants work. All of which path eventually leads me to Lancaster and what I do here. So now I'm part of the photosynthesis group, and we're really interested in improving crop photosynthesis, or at least understanding what the inefficiencies might be in terms of the photosynthetic system. A lot of the work at Lancaster is focused on rubisco, which is this central enzyme that does the reaction of taking carbon dioxide out of ultimately the air and turning that into something through the Calvin cycle, sugars that will be part of the plant's metabolism and its body and contribute to all of the things that it needs to do in terms of reproduction. And in our case, generating us food. Often we're eating those reproductive parts. So the crops that I've worked on since I've been here include wheat, which is what's pictured at the top here. I've worked with some brassicas as well. So that's a close-up of a brassica leaf, bottom left. And most of our work at the moment is focused on cowpea, which you can see sitting in a really nice piece of equipment that is my kind of bread and butter for my work, which is called a LEC 6800. And that is a system that lets me measure the gas exchange of the leaf, how much water is coming out, how much CO2 is going in, and also characterize some of the photosynthetic reactions with a thing called panfluorimetry, where we can get a sense for how efficiently light is being used by the chloroplasts of the plant in their business of fixing carbon dioxide. And just on the right-hand side is a sort of indication of the key thing that we're interested in about these crops and understanding their, in terms of understanding their efficiency, which is that photosynthesis is not something that just happens in a static environment. And actually, this is something that through my entire career I've been interested in terms of how diurnal cycles are affecting plants and how they're regulating to get around that. What we're interested in now is how, as the sun moves around and other leaves in a crop canopy get between a particular leaf and their source of energy, what's that doing to the photosynthetic process, and how does the photosynthetic process have to adapt to that? So we have a diagram there where we have photosynthesis in bright light that's then transitioning into photosynthesis in shade and transitioning again out of that. And each of those steps is something that requires a lot of coordination of a series of different elements of biochemistry that take time and are therefore a little bit inefficient. And we are trying to work out just how inefficient that is and how much we can tweak that to try and make crops do a little bit more with the energy that they're getting. Yeah, I think that's probably enough for people to go off. So
thank you very much for that.
Yeah. Would you like me to stop sharing now, or are we going to do questions now, or questions once we've done everybody?
I was thinking questions for both of you together afterwards.
Okay,
if that's
so
all right.
I will stop sharing so that Catherine can have a go.
So Catherine Walsh is one of our postgraduate researchers here at Lancaster Environment Centre with the Waitrose postgraduate research program. I will hand over to you.
Thank you. Right, let me try and share. Okay, so thank you for inviting me. I'm here to give a student's perspective. I just thought I'd talk about why I find plants fascinating and just explain a bit about what my PhD project's about. So I first got interested in plants as a child, just growing sunflowers in the backyard. And then I was fortunate enough to go to a high school where we did rural science, environmental science, and horticulture. So from there, I went to ISCO. I did an undergrad in garden design. I then had 3 children and then decided, actually, I don't like garden design. So I went back and I did another undergrad in horticulture and plant science. And then I came to Lancaster to do a master's in sustainable agriculture and food security, which I did part-time, but unfortunately I had a very bad horse riding accident and managed to smash my head up. So I had to take a year off because I had concussion for 10 months. And then during that time, I got an email out of the blue from the Simon Rogan group up at Cartmel, who have Longclume and other Michelin restaurants, and they gave me a job. So while my head was getting better, I went up there and did some growing for them. And from there, I got poached to another Michelin restaurant as a head grower, which gave me a lot of free rein to do some really cool stuff, which I thought I would share with you. There we go. So this is what we used to do up there. This is the microgreen greenhouse. We used to use these plants for garnishes and on the bar. They are just like regular crops, but we've just grown to a small size, tightly packed. And they're great because they've got like 10 times the nutrients in than a regular size one. They're quite tricky because everything had to be regular and uniform. And then at the bottom there, we've got a little photo of the polytunnel, that which I'm thinking must have been winter because we've got a lot of mustard and broad beans in there. And then it allowed me to play around a lot. I've called this slide novel technique, so that's not quite true. That one on the left, that's a hotbed. So that was first seen in the market gardens in Paris in the 19th century. And what it is, it's a layer of horse muck with a layer of soil on top. And then I used to grow salad in there, covered with a bubble wrap to keep the heat in. So as the horse muck breaks down, it warms the soil. But what you can't see is that was February with like an inch of snow on the outside of it. But it was a great way to get salad going through the winter by bringing that method back. In the middle, that's the first successful attempt of oyster mushrooms. I used to go into the staff house and commandeer someone's bath to soak those sacs in, so they're in like a sugar solution. I think it was like a conifer bark chip in there that they liked. And on the right there, that was my youngest son, where we went birch tree tapping to get the birch sap, which you needed quite a lot. I think you needed something like 20 litres to get about 200 ml of syrup or something. So it's a lot of carting 5-litre containers around. So from there, I went on to apply for PhDs. It took me a while because I couldn't find one that I actually wanted to do. And then eventually one came up here. So this is my PhD project that I'm supervised by Marge Lundgren and Mike Roberts. It's a Waitrose CTP funded one, and it's optimising yields and nutrition in wild rocket. So what's cool about wild rocket is it's the only crop in commercial production using C2 photosynthesis, which is a rare type of photosynthesis used by around 56 species. And it splits photorespiration across 2 cell types. And by doing that, it optimizes the process and can reclaim CO2 that would have been previously lost by a regular C3 plant. So because it's quite a rare type, I thought I'd just show you what that looks like. So here's a C3 plant, and we see the photorespiration cycle. So this is when rubisco in the chloroplast will fix oxygen instead of CO2 and forms glycine. And when glycine decarboxylase enzyme works in the mitochondria, we see CO2 and ammonia released, and it acts as a cycle. And under ambient conditions, our C2 crop's going to be doing that as well. But what it does when the photorespiration kicks in, which is the hot, dry conditions, is we see that same process split across 2 cell types. So all the glycine decarboxylase is confined to the bundle sheath, which traps the CO2 and acts as a simple carbon concentration mechanism. And the ammonia that's also released, we're not too sure what happens to that. And part of my PhD is keeping an eye out to see what the data is showing us as to where that goes. So what we do know about rocket is it's got a high amount of nutrients in it. You only need a small amount to provide you with your daily recommended intake of some of these compounds. It's high in antioxidants from the vitamin C and the flavonols. And to get all these compounds. It's a combination of carbon, sulfur, nitrogen and sulfur. And I'm looking, as part of my research, how these interact and how these are going to convey those nutrients under climate change. So as climate change advances, because it's a poorly understood mode of photosynthesis, by understanding how it works, we can see if it does maintain its nutrient stability as well as its yield and stress tolerance. And from there, we can see how we can engineer it into a C3 crop. So they might do the same. So that's it. That's just an overview of what I do here at Lancaster. Thank you for listening. I'd like to acknowledge my supervisors, Marge Lundgren and Mike Roberts. And if you have any questions on either rocket or C2 photosynthesis, then I'm happy to try and answer them. Thank you.
Thank you very much, Catherine. So we go to questions. If anyone has any questions, you can either do the sticking up your virtual hand or writing in the chat. Either of those is absolutely fine. And that can be questions for Catherine or Sam on their research or generally on their fascination for plants, whichever. While people are writing, I'm going to take chair's prerogative and ask a few. Catherine, I was really interested in how you said you'd done sort of rural science, environmental science and horticulture at school. That's got to be pretty unusual that those things are covered, right?
Yeah, I was really lucky there. I think probably wouldn't be here now if we hadn't done those things at school. I'd probably end up doing something else. But that was just in Lancaster. That was at Ripley.
Ah, Ripley has a farm, doesn't it?
Yeah.
I've walked past the sheep.
Yeah.
Sam, what was your sort of introduction to plant science? Was it that, you know, you did a bit of it at school, but I remember it being quite dull when I'd learned it at school?
I mean, I certainly learned a version of the metabolic network of photosynthesis and respiration when I was at school. I think the place where I got an enthusiasm for it was doing ecophysiology courses at university. That said, I mean, I grew up in a really rural environment. I was in Northumberland myself, and so I was often around forested areas and moorlands, and you know, thinking about why things are where they are has always been part of what was interesting to me. And my degree was mostly an ecology degree for that reason, that I was interested in, you know, looking at landscapes. We used to come as a school on field trips to the Lake District, and my sort of basic interest in natural history, I think, came from spending a summer living here on the other side, actually not far from Lundgren, over by Grange, when I was about 7 years old, going up on the hills there and seeing the ponds full of all kinds of amazing things on the top of the fells, and just falling in love with nature, really. But then there is a spot in Northumberland that I like to go to when I get a chance to go up there, where there's a bit of forestry we used to walk around as a family when I was a kid, that has quite a good grove of sequoia growing in it. And that was a place that as a teenager I used to go to, and it just, the sense that plants have a direct effect on your immediate environment in a place like that is really clear, because the temperature and the humidity and everything else changes really dramatically when you go into those groves. And so that was another thing that sort of prompted me to be interested in plants. But it took me getting to late in my undergraduate to have sort of really understood how you could study them and think about them.
So you started with aphids in your research. Did you come at it from an entomology direction, or I don't know, a food security sort of direction, or just because it was the subject on offer?
It was a return to, it was an interest in food production. So that studentship was, I think, one of the last that DEFRA directly funded, and it was done through ADAS as well. So we were working directly with a team at ADAS on that. And I had gone to that having kind of gone and done a short period where I was working in an environmental monitoring kind of role, which I could see just looked like paperwork in the future. I was going to go from doing something in the field to doing paperwork at a desk, and I didn't really fancy that at the time. So I wanted to go and do something where I could do more exploring. And I'd got to where I was in terms of ecology, in terms of a lot of symbiosis and thinking about how organisms interact. And so I came into that stuff around with the aphids around, you know, what is it about aphids interacting with potatoes that affects their ecology, basically. Yeah, so it wasn't entomology, but I learned some while I was there.
Catherine, you've obviously got a food production background in there, working for food production and Michelin star restaurants. So is that something that you hope your research will be applied back into, or is your current research a bit more fundamental? That's the word I was after.
I think it's fundamental regarding the metabolism, but then I'm not 100% convinced that there's one C2 phenotype, and there could be many. So I'm sort of researching specifically rocket as the only crop species. So I'm not 100% certain if that can then be applied to the others or not. It's difficult when I'm just working with one species.
I'm going to therefore ask the ignorant non-plant scientist question. We've got C2, C3 and C4 photosynthesis in here. What?
Well, C3 is the common one. I think that's over 97% of plants use it, but it's affecting our crop yields. Once the photorespiration kicks in when it's hot and dry, so you get around a 30% flux through that. So then you see a yield decline. So the C4 is obviously a high-performing photosynthetic version. Well, not C3, but that's the holy grail, I guess, of photosynthesis. And C2 is like a halfway evolutionary point, but it's also a stable state in its own right, which is perhaps a good way to explain it. I don't know.
I think my perspective on with the question you're asking to a carbon concentrating mechanisms person is about carbon concentrating mechanisms, is that one of the things that we can learn from plants about is actually a lot of stuff about deep geological time and how the earth has changed. Without the photosynthesis that plants do, we wouldn't have an oxygenic environment. I mean, that was probably originally bacteria doing that, but there's another element, which is that plants represent some link to this idea of what we are as organisms and how different sets of organisms have come about through different combinations of cell types. But when you look at CO2 in the past, the driver for things like C4 and C2 emerging, which are relatively recent, is a geological trend in CO2 levels, that there was a superabundance of CO2 at the time when plants first came onto the land. And that's something that changed with time to the point where it was quite hard to get hold of CO2. So you get these things as carbon concentrating mechanisms that are a system for trying to deal with that. And now we're in a stage where scientists are looking at taking inspiration from that to try and improve plants that didn't make that improvement themselves.
So would that be an improvement again back to yield and crop production, or is that could that be used as a sucking more CO2 out of the atmosphere to help fight climate change sort of development?
I think you're probably talking about different scales at some level there. CO2 coming out of the atmosphere has to go somewhere. And so that's going to have to go into some pool that's then non-labile. With a lot of what we're doing with crops, there is some sequestration going to happen there, but I'm not sure that we're talking about timescales where we would be addressing the climate crisis by improving photosynthesis. Yeah. There may be others in the audience who would be better or as equipped to answer that question as I am.
Speaking of the audience, we've got a question in the chat from Andrew Slater. I don't know if you guys can see that.
Yeah. Do you want to take that, Catherine?
No.
Okay.
No,
Well, I could maybe take that. Oh, is that the answer?
Yeah. Sorry, no, CAM isn't related to C2 as far as I know. I think CAM is like a time separation, whereas C2 is a spatial separation. And yeah, C2 is more in similarity to C4.
Yeah, the bit that's the intermediate in CAM is a different kind of compound as well. So the name CAM is crustacean acid metabolism, and so it's an organic acid that's being stored as a pool of carbon that can be accessed later, which is not an amino acid, which is my understanding of, because your glycine that's involved in the C2 is that. So it's using a different branch of biochemistry to come to a sort of similar endpoint in terms of being a carbon pump of some sort.
We've got another less technical question, which I guess will go to both of you. So maybe to Catherine first. Hannah Plant has asked, what made you decide to do a PhD?
Well, it probably goes back to when I was here as a master's student, and there was a couple of PhDs I wanted to apply for, and then, as I said, I had an accident and had concussion for 9 months, and so I just couldn't do it then. And then I thought, if I don't do it now, I'm going to be too old. I'm old enough now to be doing it.
I don't think you're ever too old to do a PhD. You might be too impatient.
So what was it about? Was it that particular subject then, Catherine, that, you know, the particular questions the PhD was asking, or did you want to do a PhD to do 3 years of research?
I think because it was on Rocket as well, I love a good salad crop, and I'd spent years working on trying to intensify flavours for all the chefs I was working with, and there's an element of that in working with Rocket because it's known for its peppery taste, but then that brings with it a lot more nutrients and things. So I'm just sort of looking into the biochemistry to keep the nutrition stability there. It just sort of followed on, really, a more in-depth version of what I was already doing.
What about you, Sam? What took you down the PhD route?
I didn't want to get strapped to a desk. I mean, I think I answered that in a previous question.
So you're sure you're not strapped to a desk anymore?
I was always curious about biology, and I went to do a biology degree because I was excited about understanding evolution and ecology, and it wasn't immediately something I was doing with that when I came out of my degree, and the obvious thing to do was to keep learning as far as I was concerned. So.
Yeah, that's a very good answer. It's definitely 3 or 4 more years of learning, and then more learning after that.
Yeah.
Are you hoping to stay in the research world, Catherine?
I've not thought that far ahead, actually.
Fair enough. We'll move on. Does anyone else in the audience have a question that they want to ask, either a general one or a research specific one? We've got silence at the moment. What's your favorite bit about your day job, you take a day of your current work. What's your favorite bit? I'll go to you, Sam.
I mean, it's always been about problem solving, I think, for me. Like, if I get a really thorny problem that I can spend a few hours worrying about, I really enjoy that. And then there's a tremendous reward, particularly with getting an experiment design worked out, where you finally worked out how to answer a question, and then you think you've got it, and then it breaks. But there's a sort of thing that you're constantly chasing fixes to things, and I quite like fixing things. So.
What about you, Catherine?
Yeah, I'd agree with that. I like the problem solving, definitely. I don't know. I like when I'm reading papers, trying to pick out what's unusual or what hasn't been followed up in like other papers, and then having a go myself. And I love doing diagrams. I could sit all day just drawing biochemistry diagrams.
That's a transferable skill no one talks about. I like that. Oh, we've got an excellent question in the chat from Ingrid. What's your favourite plant? And you're not allowed to say rocket, Catherine. So I'll go to Catherine first and put you on the spot.
Oh, that's a really good question. Thank you, Ingrid. I quite like probably wood sorrel, actually, because it's everywhere, and I'm often picking at it when I'm walking my dog, and it just tastes so sharp. It's like sucking lemons, isn't it? I do like that, and it looks so pretty as well, actually. Yeah.
It waves at you as well.
Yeah.
Yeah.
That's really hard, just picking one.
Do you have a favourite, Sam?
Yeah, I think I'm always excited when I see a sundew, because they're such, apart from anything else, whenever you see pictures of them, you see they look like they're really massive. And then the ones that you actually see in bogs in this country are, if at best, a couple of centimeters across the whole thing. And they're just little and weird and sticky. And, what's it doing? It's such an interesting little thing. And then it has a really pretty little flower that comes out of it as well, when you get a chance to see that. So they're kind of cool.
What would you recommend if somebody wanted to take up plant science, plant research? What would be the first step you think they should take? Either of you?
It sounds to me like I don't think there's any clear first step. I mean, because if we were to look at me, I went from school to university and started learning about plants. And if we look at Catherine, she did something completely different to that. She's still here doing the same thing. So, I mean, yeah, be interested.
Yeah. Yeah, that sounds good. Be interested is, curiosity is the way in, isn't it? It's the thing that keeps you going, I think. Wherever you find that curiosity, be it in taste or look or, it's a weird plant or whatever.
Yeah.
I think there's a lot of apprenticeships as well now. So you could always go down that route, see if you like it on the practical side, and then go down a more academic route to dip your toe in, maybe. Yeah.
Is there anyone in particular that you look up to, be that like sort of famous people or someone in research or something, someone that you would like to have a chat with about what you do? You ask this of ecologists and everybody just says David Attenborough, you see, so I thought it'd be interesting to ask the question to plant scientists.
I'm going to go a bit off kilter there, because I don't know that there's a plant scientist, but I think I would find it interesting to talk to Brian Cox about plants, because that version of thinking of the world in terms of a series of physical interactions and the energy from the sun and the quantum mechanics of that, and how that feeds into us having these things that live and regenerate on this planet. I find that stuff fascinating.
That's a very different perspective.
It's that sort of idea of being transported into a different way of thinking about things. I think that's really important for me. So.
Any thoughts, Catherine? It could be a chef, I suppose, if you know.
I don't know really. I'm still trying to wade through Green Planet, so probably David Attenborough's a good one for me. Well, he's still here. Shouldn't say that. I don't know really. Sorry, I was
to say your PhD supervisor.
Sure, Marge will sit there.
You have many conversations with your PhD supervisor, don't you? So she inspires you every day. That's what we're supposed to say, right?
Yes.
Oh, we've got a thumbs up from Marge. Excellent. There we go. Yeah, I mean, if there are any more questions, feel free to ask. The detail, I'll just post in the chat the link to the webpage for the rest of the interactive Fascination of Plants Day activities that our plant scientists have put together. So you can go and explore those. Fascination of Plants Day is like an international celebration of plants and plant scientists that happens I think it's sort of supposed to be every other year, but obviously it's all changed a little bit over recent times. But I know our plant scientists have been doing both in-person and online activities for a while. So it's really great. As someone with an interest in science communication, thank you to all of you, Sam and Catherine and the rest of the team, for sort of engaging with this and sharing your interest and your research. I think unless we've got any extra questions, we can take you off the spot, take the pressure off. But thank you very much for sharing with us today. I found it really inspiring, and I'm totally going to go and look up all these many forms of photosynthesis that I didn't know existed. And eat rocket. That will be my next thing to do. So thank you very much to both of you. I will just give a quick promotion to some in-person LEC seminars that are coming up next week. So these won't be online. These will be in person within the department. And hopefully we will be able to record them. So we will be sharing them on the YouTube channel afterwards. But all of the details for our LEC seminars are found on our website. And in particular, as I say, these two in-person seminars coming up next week. One is on Wednesday, looking at the assessment of impact of habitat conditions on gross primary production of forest ecosystems, with Mr. Caleb Mensah in the training rooms on Wednesday. And then on Thursday, we're looking at risk, emergency and sustainability reflections on the Brazilian context for global health, with Dr. Gabriela Marques de Guio. And that will be on Thursday in the Biology Lecture Theatre. So a quick promotion for those, but details, as I say, on the website. And thank you very much again to Sam and Catherine. Much appreciated, particularly for helping me organise this at quite short notice. So thank you very much. And I'll leave it there.
Thank you.
Thank you.